Hello everyone. In last session we saw about structure of protein. In today's session we will be seeing about Ramachandran plot. Today we are going to see about Ramachandran plot. We have to know something called torsion angle. So as we know, proteins are made up of polypeptide chains. Polypeptide chains are formed because of the presence of the peptide bond formation. The peptide bond formation occurs when one carboxyl group of the one amino acid tend to form a bond with the amino group of the another amino acid with the release of the water molecule. This process leads to the formation of the peptide bond formation. So as we look at the protein structure or polypeptide chain, we have to know there are three main bond angles. So the bond angles are formed because of the presence of the three main atoms. So we can see alpha carbon, nitrogen atom and carbonyl group. So between these atoms there will be the presence of three major bond angles. So the bond formed between alpha carbon and nitrogen is called as pi bond, and then the bond formed between alpha carbon and carbonyl group is called side bond. Finally, the last bond which is formed between carbonyl group and nitrogen is called as omega bond. The three bond formation, that is pi bond, psi bond and omega bond, plays an important role in the protein stability and also plays an important role in protein conformation. And also the 3D structure of the protein is also determined by these three bonds. So now we are going to see about torsion angle. So in order to understand about torsion angle, I am going to discuss about three main molecules, that is hydrogen molecule, water molecule and hydrogen peroxide molecule. When we look at the hydrogen molecule, in hydrogen molecule there will be two hydrogen atoms. So the hydrogen atoms which are separated by bond, or we can say the hydrogen atoms are formed or separated by some distance. So in the hydrogen molecule we are able to see some distance in the hydrogen molecule, whereas there will be no bond formation, or we can say there will be no angle formation. Now when we look at the water molecule will have three atoms, that is one hydrogen atom, another hydrogen atom and one oxygen atom. So there will be two hydrogen atom and one oxygen atom. Finally, when we look at the hydrogen peroxide molecule, there will be four atoms, that is two oxygen atom and two hydrogen atom. And when we look at the bond formation in the hydrogen peroxide, there will be three bond formations. And when we look at the angle in the hydrogen peroxide, there will be two angles. And the geometry of the hydrogen peroxide will not only determined by the bond and angle, there is something called planes. Planes also plays an important role in deciding the conformation of the hydrogen peroxide. So angle, bond and planes plays an important role in determining the conformation of the hydrogen peroxide. So angles are determined by the planes or not by lines. So this is how the 3D structure of the proteins are formed. So these angles are known as torsion angles. Now we are going to see about steric hindrance. Steric hindrance is a consequence of the steric effort. Steric effort causes the chemical reactions to slow down because of the bulkier R group presence. And when we look at the steric hindrance, it is usually manifested in the intermolecular reactions and also affects the intramolecular interaction between the molecules. When we look at the steric hindrance presence in amino acids, steric hindrance largely affects the torsion angles because amino acids tend to have a different R group molecules. R group molecule will not be in a linear range. R group will be varied for different amino acids. In those cases, the bulkier R group tend to have an torsion angle disruption when steric hindrance is affected, and the linear R group will not have a large steric hindrances because of the less bulkier groups. Now we are going to see about Ramachandran plot. Ramachandran plot was first described and discussed by G.N. Ramachandran in the year 1963. He studied about the protein stability and conformation with the help of the collagen protein. So Ramachandran plot is a visualization of the backbone of the amino acid residues, and also Ramachandran plot help us to find about the protein stability. So we can say Ramachandran plot is a computational model where pi and psi angle of the each amino acid residues or each polypeptide has been studied. And also this pi and psi angle plays an important role in determining the protein conformation for all possible amino acids. We could say that the pi and psi angles are oriented in such a way that which will help us to decide the steric hindrance of the each amino acids with the help of the Ramachandran plot. For each conformation in the Ramachandran plot, the structures were evaluated based on the close contact between the atoms. The atoms were considered as hard spheres These hard spheres were evaluated or analyzed with the help of the van der Waals radius. That is, the dimension of each atoms and their interactions were determined with the van der Waals radii. So, the atoms which are considered as a hard sphere were collide to each other, which helps to determine about the conformation of the protein. The conformation of the protein were determined with the help of the pi and psi angle. So, the pi and psi angle in the Ramachandran plot was discussed based on the steric hindrance, based on the allowed region and disallowed region. So, the structure validation and also verification has been done for all the possible amino acids in the Ramachandran plot with the help of the pi and psi angles. Now, we are going to analyze the Ramachandran plot. So, in the slide, you are able to see the Ramachandran diagram. So, in the Ramachandran plot, there were three main colors: yellow, white and red. In the x-axis, pi values were there. In the y-axis, psi values were there. The x-axis and the y-axis, the ranges were plus 180 degree to minus 180 degree. And also, there are three main colors. Three main colors has three main significance or conformations related to the protein structure and also protein stability. We are going to see one by one. First one is white color. The Ramachandran plot, there are more white regions. So, these white regions are sterically inheritance regions, or we can say sterically hindrance regions, where not all amino acids can be present in the white region. Only the amino acids which have a less steric hindrance or will have a less bulkier R group can present in the white regions. Now, after white region, we have to concentrate on two main regions, that is red region and yellow region. So, when we look at the red regions, we can say red regions are allowed conformational regions. So, it means that the most of the amino acids will be present in the red regions. We could say the amino acids whatever present in the red regions might have alpha helix conformation or beta sheet conformations. So, the red region is an allowed region in the Ramachandran plot. And finally, we have something called yellow region. Yellow region will have less steric regions, or we can say van der Waals forces acting between the atoms will be shorter in the yellow region. So, the amino acids that are present in the yellow regions are, we can say, left-handed alpha-helic regions, or the left-handed alpha-helic amino acids will be present in the yellow regions of the Ramachandran plot. So, these are the three main concepts or three main colors significance the Ramachandran plot. So, now we are going to discuss about the glycine and proline in the Ramachandran plot. So, in the slide you are able to see about the glycine Ramachandran plot in the left hand side and the proline Ramachandran plot in the right hand side. So, when we look at the glycine, as we know, glycine is a very simple amino acid, will have a side group of H atom. So, there will not be a much bulkier R group will be present in the glycine, and glycine is a non-polar amino acid. We could say glycine is more flexible than any other amino acids. So, in the Ramachandran plot, you are able to see the presence of glycine can be seen in the all the quadrant of the Ramachandran plot. When we look at the proline, proline is an aliphatic amino acid. Proline has also have a 5-membered ring, is also known as imino groups. So, the presence of imino group causes a steric hindrance, or we can say the imino group present in the proline gives the structural rigidity. Because of the structural rigidity, it cannot be present in all the regions of the Ramachandran plot, and also this structural rigidity of the proline makes the proline to be present in the turns, not in the helix or sheets. So, because of that only, glycine and proline will not be displayed in the Ramachandran plot, since glycine is more stable or more flexible, whereas proline is less flexible compared to other amino acids. Till now, we saw about Ramachandran plot, and we also saw how can we interpret and analyze Ramachandran plot. Now, we are going to see about the protein folding and protein denaturation and protein aggregation. First is, what is protein folding? So, protein folding is a process where the proteins are oriented in such a manner so that the geometry of the protein leads to the formation of the 3D structure of the protein. This 3D structure of the protein leads to the functional protein molecule, that is called protein folding. And what is protein aggregation? So, protein aggregation is another biological phenomenon where the proteins are misfolded in such a manner, they aggregate to each other, which leads to the formation of the disease in a human body. So, as we know how the protein folding is occurred. So we know the protein folding has occurred because of the presence of the various bondings that occurs in the polypeptide chain formation. So in the last class we discussed about primary structure, secondary structure, tertiary structure and quaternary structure of the protein. So these structure formation are because of the various bond formation that occurs in the polypeptide chain. So because of the presence of the hydrogen bonding, ionic bonding and intramolecular interaction between the molecules in the polypeptide chain leads to the formation of the 3D structure of the protein, or we can say this is a protein aggregation, which again leads to the formation of the new diseases in the human body. So as we discussed earlier, and again protein denaturation is the loss of the 3D structure of the protein which leads to the malfunctioning in the protein. So how this protein denaturation can occur, or what are the causes how the protein can be denatured? There are 5 major causes. One is temperature, another one is pH, another one is enzyme, another one is organic solvent, another one is detergent. By using these components and these variables, we can disrupt the particular protein. So now we are going to see various protein denaturation methods. First one is thermal denaturation. So as the name indicates, thermal denaturation will be using heat source to denature the protein. So when we use heat source to denature the protein, it will cause the intramolecular hydrogen bonding to break out. So in thermal denaturation, the hydrogen bondings will be break out so that the proteins will denature by itself. So how can we monitor this method? So we can monitor the protein denaturation. So the 5 major methods to determine the thermal denaturations are: first is absorbance. Absorbance can be measured using calorimetry or UV spectrometry. And then enzyme activity. Enzyme activity can be determined using some assays. And then circular dichroism is one of the light spectroscopic technique which use the measurement of the difference in absorbance in the left and right polarized light. And then anisotropy. Anisotropy is the property of the substance that differs or varies in the physical properties with varied molecular axis. And then finally fluorescence method. In fluorescence method, we can use fluorescent compound or we can tag fluorescent compound with protein to determine the protein denaturation. So next method is using pH. We can use pH to denature the protein. So before going to see about pH, we should know something called isoelectric point. So isoelectric point is the point where the protein's pH will be neutral. We can say that in that point, the positive and negative charge of the protein will get neutral. This point is known as isoelectric point. So as we know, the pH ranges from 1 to 14. So the pH below 7 is acidic in nature and pH above 7 is basic in nature. If the pH is greater than Pi, then the protein will be negatively charged. Whereas if the pH is below the Pi, then the protein will be positively charged. If the pH is equal to Pi, then the protein will be in a neutral charge. In that point only we can precipitate or denature the protein. For example, we can precipitate casein from the milk at the pH of 4.6. Since the pH 4.6 is the point for the casein to precipitate, it is also known as isoelectric precipitation of protein. So the next method is using organic solvent. So the organic solvents denature the protein with the help of the hydrophobic interaction, or it will disrupt the hydrophobic interaction that occurs in the protein. So we can say when we use organic solvents, it will make a bonding within the organic solvent and protein R chain group, so that it will disrupt the protein interaction. So by this way, the organic solvent will disrupt the protein, and also it will increase the hydrophobicity and it will decrease the water quality. By that way, this can be done with the help of the low temperature. So protein misfolding or protein aggregation leads to different diseases like trans-tyrosine amyloidosis, Parkinson's disease, Alzheimer's disease and mad cow disease. For example, in trans-tyrosine amyloidosis, trans-tyrosine protein aggregate to each other leads to the formation of this disease. Whereas in Alzheimer's disease, amyloid beta protein aggregate in that case, so that it leads to the formation of the Alzheimer's disease. These are some of the example for the protein malfunctioning diseases. There are so many diseases that are occurring because of the protein misfolding. Today we came to the end of the session. Hope you all understand about that. And in this session we saw about torsion angle, Ramachandran plot, protein folding and protein misfolding diseases. In the next session we will be discussing about methods of protein structure prediction. Thank you all.